Good morning, everyone. My name is Maren Friesen, and I'm an associate professor at Washington State University. And today I'm going to be discussing the question, can we replace synthetic nitrogen with microbes? So the air that you and I are all breathing consists of 78% nitrogen gas. However, this nitrogen is as unavailable to us and the plants that we're trying to grow as water is to this sailor on the ocean. And the reason for this is that the nitrogen-nitrogen triple bond is extremely difficult to break. There are only 3 processes that can accomplish this. The first is lightning, which fixes 5 million tons of nitrogen per year globally. The second is biological nitrogen fixation, which is carried out by this fascinating enzyme called nitrogenase. In natural ecosystems, biological nitrogen fixation contributes 58 million tons of nitrogen per year. And in agricultural ecosystems, it contributes 60 million tons of nitrogen per year. And then finally, we have the Haber-Bosch process, which combines nitrogen and hydrogen gases together at high temperature and pressure, requiring very high inputs of energy to produce ammonium. And this results in the fixation of a whopping 120 million tons of atmospheric nitrogen per year. So the invention, industrialization, and widespread adoption during the Green Revolution of synthetic nitrogen fertilizers has changed our world. Some estimate that close to half of the people alive today exist because of nitrogen fertilizers. And the overuse of fertilizers can have environmental impacts in aquatic systems through runoff. So the nitrogen cycle occurs primarily in the soil, where we have 3 main pools: nitrate, ammonium, and soil organic matter. And transformations between these pools are conducted almost exclusively by microbes. So nitrogen fixation converts nitrogen gas into ammonium, nitrification converts ammonium into nitrate, and finally denitrification converts nitrate into nitrogen gas, or sometimes nitrous oxide. And when coupled with the carbon cycle, these nitrogen transformations fuel microbial as well as plant growth, resulting in the production of organic matter over longer timescales. So one research question that I'm really interested in is how these 2 cycles are coupled over different physiological, ecological, and evolutionary timescales. So when we add synthetic nitrogen fertilizers to these systems, there's an immediate downregulation by nitrogen fixers of fixation rates because it's inhibited by its end product, ammonium. There's also increases of nitrification and denitrification. And we can interrogate the microbial population responses to nitrogen addition by looking at the abundance of functional genes responsible for these different transformations. So interestingly, we see no consistent effect on the population of nitrogen-fixing bacteria over a meta-analysis of many different studies in agricultural ecosystems. In contrast, both nitrification and denitrification show marked increases in microbial populations in response to fertilization. In particular, ammonia oxidizing bacteria respond by increasing in prevalence by 313%. So here is a really cool sneak preview of a visualization that Carol McFarland is working on with funding from Western SARE. The project is called Visualizing Microbial Agroecology. And so this is a screen grab of a 3-dimensional animation showing the transformation of ammonium into nitrate by ammonia oxidizing bacteria. There will be Nitrospira and Nitrosomus here. So stay tuned for the release of this and other videos. For the remainder of my time, I'm going to focus on nitrogen fixation because this is the largest lever that we have in order to reduce inputs of synthetic nitrogen. So nitrogen fixation is carried out by diverse bacteria and archaea that fix nitrogen using a suite of different types of metabolism, some aerobic, some anaerobic. And as shown by this cartoon bacteria, Thor of the soil, nitrogen fixation is a highly energy intensive process. And so every time Thor uses their nitrogenase hammer to cleave a nitrogen-nitrogen triple bond, it actually requires 16 molecules, lightning bolts of ATP. Also important for understanding the ecology of these organisms is understanding that nitrogenase is strongly inhibited by oxygen. So nitrogen fixers can be symbiotic, in which they're housed inside of carefully regulated specialized plant tissues. They can also be associative, either in or in very close proximity to plant roots or shoots, or they can be free living in soil, where they have a much higher influence by the surrounding environment. So to understand the controls on nitrogen fixation, we need to know who is there, and we need to know the environments that they are experiencing. So this table sort of simplifies 3 of the main controls on nitrogen fixation across the range of potential fixers that might be present. So the first important environmental consideration is carbon availability. So this translates directly into the energy that microbes have to fuel the nitrogenase reaction. The second is oxygen protection, which protects nitrogenase from the toxicity of oxygen. And then finally, nitrogen transfer. So if nitrogen is transferred away from the microbes, then they can be persuaded to fix additional nitrogen. So in symbiotic systems, these conditions are all met very, very strongly. And in these systems, we can see from between 50 to almost 500 kilograms of nitrogen fixed per hectare per year. In associative systems, the conditions are typically slightly less optimal, with correspondingly lower fixation rates. And finally, free living nitrogen fixers experience the least favorable conditions for fixation, and despite this, can sometimes fix up to 80 kilograms of nitrogen per hectare per year. So still can be appreciable, but highly variable. So I'm going to take us through some examples of nitrogen fixers, the very studied of which is the legume rhizobia symbiosis. So rhizobia are soil bacteria that use specialized chemical signals to colonize compatible legume host roots. Their host then develops a specialized root nodule where the rhizobia fix nitrogen. And in fact, rhizobia only fix nitrogen in nodules, exemplifying how specialized this interaction is. Legume nodules contain a molecule produced by the plant called leg hemoglobin, and this binds oxygen, just like the hemoglobin in our blood does. So this is why if you cut open a healthy nodule, it'll be pink inside. That's the leg hemoglobin interacting with oxygen. And legumes have been used in agriculture for millennia. So things like grain legumes, cover crops, etc. You guys are all familiar with legumes. Another really interesting nitrogen fixing mutualism is that between azolla and cyanobacteria. So the cyanobacteria, in this case, disperse with their little water fern host, these azolla here. They're contained in the spores, which is really cool. And together, these partners rapidly grow in aquatic habitats, and they're widely used as a green manure in rice paddies, both in Southeast Asia as well as in Hawaii. Another really interesting system that's been described over the last decade is a Mexican landrace of maize. So in addition to the normal roots that grow in soil, this particular landrace grows aerial roots that secrete really high amounts of mucilage. I've been told that this mucilage can be found dripping down into the soil. There's just so much of it. And it's then colonized by nitrogen fixing bacteria that are able to provide fixed nitrogen to the corn. Another really well-studied system is that of sugarcane. And so in Brazil, it's often grown without any added nitrogen, which is really, really interesting. And there's been a number of microbiome studies in these systems that have found extremely diverse nitrogen fixing communities, both on the surface as well as inside the tissues of the sugarcane, both above ground and below ground. So it's unclear if there's a single bacteria that's providing most of the nitrogen, or if it's really happening in this community context. And so here is some data from a study demonstrating that fertilization with molybdenum, one of the cofactors of the nitrogenase enzyme, has as large or larger effects on biomass yield than fertilization with synthetic nitrogen. And furthermore, when there's fertilization with both molybdenum and nitrogen, there's no additional benefit. So this suggests that they are impacting plant growth through the same mechanism, i.e. the provision of nitrogen. Perennial grasses can also benefit from nitrogen fixation. So this has implications for things like CRP and other perennial systems. So this is a switchgrass system that I've been working on with funding from the Department of Energy in collaboration with folks at Michigan State University. And what we found is that the potential nitrogen fixation in these plots varies dramatically, both over seasons as well as between years. We have characterized diverse nitrogen fixers present in the rhizosphere soil. And by collecting large amounts of environmental metadata, we were able to find that having higher soil moisture is the best predictor of nitrogen fixation rate in this system. So last but not least, we have the free living nitrogen fixers that can be found just about anywhere. These are numerous, they're extremely diverse. And I'm part of a project along with collaborators at Clark University and the ARS here in Pullman, where we're investigating nitrogen fixing bacteria at the Cook Farm Altar. And this plot just shows the diversity of potential nitrogen fixing taxa present at this site. So although currently only bacteria and archaea can fix atmospheric nitrogen, there's multiple international projects that are trying to change this by engineering nitrogen fixation directly into cereals. So there's a group in Spain that is targeting the mitochondria for expression of nitrogenase. There's another group based at Stanford that has been working on engineering cyanobacteria into a new organelle called a nitroplast. And then there's multiple teams that are engineering signaling pathways from the legume rhizobia symbiosis into other plants in order for them to associate more strongly with nitrogen fixing bacteria. So some of the key challenges to understanding and being able to apply biological nitrogen fixation is that there is not currently an in situ way of measuring nitrogen fixation rate in real time. So we do have stable isotope methods such as 50N, but these are necessarily integrated over typically longer periods of time. In addition, the potential rates that we can measure depend really strongly on the measurement conditions, particularly the types of carbon that are added, as well as the oxygen. These rates can vary over time, so even if nitrogen fixers are present, we know that they're not always fixing nitrogen. And finally, there is a really high diversity of nitrogen fixing organisms and types of metabolism, which is very fascinating, but makes it challenging to generalize across systems. So despite this, there are a number of practices that we can use to try to promote nitrogen fixation in agricultural systems. And it's important to note here that these are not going to be appropriate for every circumstance. And furthermore, their effectiveness will vary based on your particular conditions. So the first we saw in the sugarcane example, and so that's fertilizing with metal cofactors of nitrogenase. So molybdenum containing nitrogenase is the most common isoform. There are also forms of nitrogenase that contain vanadium instead of molybdenum, and those that contain only iron. But molybdenum is hands down the most prevalent and has the highest fixation rates. So molybdenum, however, is hard to measure in soil. So to know if you're molybdenum limited, you basically need to do a fertilization test. And unfortunately for us out in Eastern Washington, molybdenum is less available in acid soils. So pH is a key covariate here. Iron is also a critical cofactor for nitrogenase, and this can be abundant in soils, but not always available. And it tends to have lower availability in alkaline soils. So next, there's the approach of reducing tillage, which from the perspective of nitrogen fixation is most important for the promotion of soil aggregates. So what these aggregates do is provide low oxygen or even no oxygen anaerobic habitats for free living nitrogen fixers. And nitrogen fixers can themselves secrete extracellular polymers that both protect them against oxygen and that help form these aggregates. So co-benefits of reducing tillage include reduced erosion as well as enhanced water holding capacity. So we saw previously that adding synthetic nitrogen inhibits nitrogen fixation activity. And in addition to this, legumes are known to downregulate nodulation in response to nitrogen, particularly nitrate. However, when organic nitrogen is added, we found in our meta-analysis that nitrogen fixers actually tend to increase in the system. And we suspect that this could be because nitrogen fixers, since they have access to this large atmospheric pool of nitrogen, are able to invest in these costly extracellular enzymes that then are excreted from the cell and can degrade organic nitrogen so that it's available both to them as well as to plants and others in the soil. So the most tried and true method of fostering nitrogen fixation is, of course, to plant legumes, which have very high rates of nitrogen fixation. And these can be used in rotation, things like garbs, peas, and lentils. They can be really great cover crops, things like clovers, which has erosion co-benefits. They can be used as forage, things like alfalfa, or they can be used as intercrops, such as pea and canola or piola. And because legumes typically are relatively specialized on their rhizobia, it's usually important to make sure that you have compatible rhizobia present in your system before planting a lot of legumes. And the rhizobia partners are typically well known and can be very easily inoculated either as seeds or peats, or there's a variety of different methods there. So a emerging and pretty interesting potential mechanism for harnessing biological nitrogen fixation is compost teas and biofertilizers. So it seems from my reading of the literature that there is definitely more research needed here, but these teas can definitely contain free living nitrogen fixers, as has been shown by some sequencing studies. So the thing that I'm curious about is whether we can enhance nitrogen fixation during the brewing process to increase the nutrient content of the teas before they're applied onto fields. Compost teas can also be a source of beneficial microbes themselves, so they can function as inoculants too. And then, of course, there's commercial microbial inoculants. So this is a really hot area right now. Biofertilizers are a large and fast-growing market. Lots of industry interest here. So in North America, the market is almost $500 million, with large markets other parts of the world as well. And of the products available currently, 79% of those are nitrogen fixers, and another 14% are phosphate solubilizers. That I've seen for microbial inoculants suggests that sometimes they are super effective, and other times they are either ineffective and sometimes full effects. And it's currently not really well understood why they succeed sometimes and not other times. So in addition to this variability, there's some concerns that myself and some colleagues raised a couple of years ago, that these inoculants could potentially have unintended consequences if they're able to escape into neighboring ecosystems. We argue that utilizing more highly specialized microbes reduces the risk of escape. So for example, rhizobia have been inoculated into ecosystems for over 100 years. And finally, monitoring these microbes is key to understanding their ecology and potential impacts. To conclude, I think that there is a lot of promise for reducing synthetic nitrogen fertilizer with microbes, but that it's going to take diverse and really locally customized approaches. There's no silver bullets here. I think that we can learn a lot from the nitrogen fixers that associate with legumes, azolla, and grasses. And a guiding principle is to manage the ecology of these systems to promote the growth as well as functioning of either existing or inoculated nitrogen fixers. Complementary to this is engineering or breeding for higher levels of nitrogen fixation in both plants and microbes, as well as careful experimentation with inoculants and compost teas to understand when and how they're working. And since this is such an active and really exciting research area, I think that the largest gains will come from co-innovation between farmers and researchers through forums such as the Farmers Network. And with that, I'd like to acknowledge various funding agencies for supporting my lab's research over the years, my current lab members, and alumni from my lab. And I'd also like to thank all of you for joining us this morning.
Wonderful. Thank you, Maren. As a reminder to our attendees, please answer the one question survey. Please go to the Engage tab above the video stream to answer that question, and then click back on the video tab to come back to the video. So Maren, we do have some questions. There are a couple of questions about molybdenum. So as you mentioned, molybdenum can be difficult to analyze in soils, or at least analyze with high precision. So there's a question about, is it better to measure molybdenum using a tissue sample? Or can you say more about the fertilizer testing that you mentioned as maybe being superior to a regular soil analysis?
Sure, yeah, a great question. Yeah, so I don't have personal experience with SAP analysis. I know that it is possible to get molybdenum levels in your plant using SAP analysis. However, it's important to keep in mind that the molybdenum in your plant reflects how much molybdenum the plants are able to take up and process. And so if your plant is molybdenum deficient, that doesn't necessarily mean that microbes in the soil will be molybdenum sufficient or deficient, because they can have different strategies. However, if you know that you have a nitrogen fixer in your system that's functioning as an endophyte, or if it's a legume system where, the rhizobia are inside the nodules in the roots, then definitely looking at molybdenum in sap would be a great idea.
Okay, and you mentioned doing fertilizer testing. So is that something that producers can do on their own?
They would not be able to do it.
Okay. Okay.
All
right. Good. So yeah, just to bring that home, like that's something that producers can actually do on their own, is to apply molybdenum if they're concerned that that's low, and use control areas to compare to. Some other questions. Is there research on indigenous microorganisms, indigenous nitrogen fixers, and how those might interact with the amended organisms, or how effective the indigenous organisms are?
Yeah, so my lab is doing some research on that, and there's a number of other groups that are really interested in indigenous nitrogen fixers. We haven't looked directly, and then there's a long history of indigenous rhizobia that have been problematic primarily in soy production, where the indigenous taxa are often less effective than the inoculated Bradyrhizobium strains that are being utilized. And so, yeah, I mean, in terms of these associative and free living communities, it's still a little bit murky exactly who the key players are and when. Yeah, I would say that that's an active research area, and we don't have any clear principles for, on the associative side. Yeah, although, we do, as I mentioned, know a lot about the rhizobia system.
Okay, great. So the data that you showed about nitrogen fixation in the sugarcane systems is really impressive and exciting. So why can't we do that here, or why aren't we doing that here with North American crops?
That's a great question. I wish I knew the answer to that. And I mean, that's one of the things that, I'd really love to work with people on, how we can translate some of these systems into our crops.
Okay, yeah. Or is there a way that we can utilize the nitrogen fixing bacteria to make commercial fertilizer, maybe outside of the soil?
Mm-hmm. Yeah, I mean, yeah, you and I, Lynn, have talked about compost tea, and I think there's definitely a lot of potential there. Either things like azotobacter, that's a heterotrophic, really fast growing, oxygen tolerant bacteria. We grow flasks of it in my lab all the time for experiments. There's not really any reason that you couldn't just brew up a big tank of azotobacter. Or, if you were able to find the right conditions, there's cyanobacteria, which are, really cool because they perform both photosynthesis as well as nitrogen fixation by themselves. So you wouldn't even need to be adding carbon to that system, and you would just be getting, lots of organic nitrogen as well as carbon. So I think there's a lot of potential for those approaches. Yeah, and the fact that they can be done on farm is also really exciting.
Mm-hmm. Great. Yes, I look forward to pursuing that more with you. Great. Thank you so much, Dr. Friesen, our last speaker for today.